Now, this afternoon, this late morning, we're going to talk about absorption and the physics of absorption, and we'll talk a little bit about the materials in the ocean that absorb. So we talked about, we're going to talk about the electromagnetic spectrum again, and what absorption is, and what the major absorbers are. And then tomorrow we'll talk a little bit about how we measure absorption, because we'll be actually measuring absorption in the lab. But we'll talk about some of the details in the, I think Emmanuel will be talking about that with me. Okay, so electromagnetic radiation. When you have a charged particle, it's a dipole, and as it oscillates between plus and minus, it creates an electric field, right? And so you can think about, so this is the intensity of the electric field, and each one of these lines is a vector, and how it oscillates. And you can think about this as a wave traveling in space, or if you were in one point in one location, you could think about it as oscillation as a function of time, right? So when a charged particle moves, it creates a magnetic field, and you will see it either named B or H, depending upon which book and what decade that book was written. But these fields, electromagnetic fields, oscillate, and the energy propagates along a direction. So in this case, it'll be propagating along this axis. And you can follow the right-hand rule for E cross B, and that tells you the direction of propagation, right? Reminding you from physics. And we can think about this electromagnetic radiation in terms of it has a range of oscillation frequencies, how frequent those oscillations occur. And that's described by the electromagnetic spectrum. And so you probably all have seen some form of this where you can think of the wavelength of the energy is increasing this way. So if we're in the visible part of the spectrum from blue to red, or from gamma rays to microwaves, or radio waves. And you can also think about it in terms of frequency. And so, of course, the shorter wavelengths have the highest energy. So ultraviolet photons or ultraviolet frequencies have higher energy than, say, red wavelengths or red frequencies. Okay, so we're going to mostly be concerned with the visible spectrum in this class. So what is actually happening when we have absorption? So since the EMR is propagating, when materials are absorbing that energy, they're absorbing radiation, they're absorbing energy, and different things happen to those compounds or molecules when they absorb electromagnetic radiation, depending upon what the energy is. And so if you think about the energy associated with different parts of the spectrum, the energy that's given by this equation here, where this is Planck's constant, speed of light, and the wavelength of light. So since the wavelength is in the denominator, that shows you that there's more energy at shorter wavelengths, UV, less energy at longer wavelengths, red or infrared. And what happens to the molecule depends upon the wavelength, because it depends upon the energy that's being absorbed. So here's the spectrum again, from gamma rays to microwave. And I've outlined the visible light here. So let's think about what happens when a molecule absorbs microwaves. What happens is the molecule can rotate. There's enough energy in the packets of energy that are being absorbed to rotate the molecule. And so if you're rotating the molecule, well, we use microwaves to heat food up, right? Okay. If you get into the infrared, and molecules absorb infrared radiation, it has a little bit higher energy. And so the molecules can actually start to vibrate, right? And so this is a water molecule. And if this, say if this is the oxygen and these are the two hydrogens, you can get vibration of the bonds like this, or like this, or you can actually get some flexure like this. Okay? That's what happens to the molecule, water, when it absorbs infrared energy. When you start getting into the visible and into the ultraviolet, and even a little bit into this extreme ultraviolet, those packets of energy are a lot stronger. And so what happens to the molecule is that you can get what's called an electron transition. So an electron that might be in some shell absorbs enough energy that it actually moves it to a higher energy state or electronic state. It takes a lot of energy to move electrons. It turns out it takes the energy equivalent of an ultraviolet or visible photon to do that. So anytime a molecule is absorbing light, that's what's happening to it. Electrons are moving. Now, the amount of energy to go from here to here for this made-up molecule is quantized. There's only one allowable chunk of energy that can make that happen. So this particular made-up molecule needs to absorb exactly the amount of energy that can move this electron to a new state. If there's more energy, it won't do it. If there's less energy, it won't do it. That's the quantized part. So for each molecule, there's a certain amount of energy required to move an electron from one state to the next. Okay, so there'll be slightly different amounts of energy along this part of the spectrum, and it will be in one of those. Maybe it's the blue photon, maybe it's a red photon that's sufficient. It depends on the molecule. Okay. X-rays actually cause electrons to leave, creating an ion. It's a much higher energy state. And gamma rays are sufficient that when absorbed, they will change the nuclear configuration of the molecule. That's why they're so damaging. Okay. So absorption changes molecules, changes atoms and changes molecules. So let's do an example. Let's think about, I'm going to use an example of chlorophyll. So we're going to think about the amount of energy that's required to move an electron from the ground state to a first excited state, or like electronic state, or maybe even a second electronic state. So if we think about, here's energy on this diagram. If you start the molecule in a ground state and it absorbs a blue photon, that's a lot of energy. It can take this ground state molecule into a second level electronic state. That same molecule, chlorophyll, which is interesting because it actually has two quantized states, or it can absorb energy in two different intensities, two different amounts of energy. It can also absorb a red photon, and that will take it into this second electronic state. It's a little bit less energy, but it takes an electron to a different state. So you have two allowable states within the same molecule, which is really unusual. Very few molecules have two different electronic states. It's one of the amazing things about chlorophyll. It allows for wonderful things to happen with photosynthesis, which Ivana will talk about later. So we can have this same molecule with either its second or third electronic state, depending upon which energy it has absorbed. What that means is that if you think about it in terms of an absorption spectrum, there's an absorption coefficient at the wavelength between these two electronic states. So whatever this distance is in terms of energy, that is a 1-to-1 correspondence to the wavelength because of hc over lambda. So if the molecule has this allowable quantized energy difference, that energy defines the wavelength of absorption allowed by the molecule. In this case, about 440 nanometers. Yeah?
How specific is that? Like, could it be 1 nanometer off?
So it depends. It's a good question. It depends on the state of the molecule. So if you took a chlorophyll molecule and you dissolved it in acetone, the solvent will change the structure of the molecule a little bit. So maybe instead of being 440, maybe it's 430, because the bonds of the molecule are different, which would change, maybe it would make it a little bit higher energy state here, compared to if you, say, put it in methanol. or if the molecule was attached to a protein, or if the molecule was attached to a protein in a thylakoid membrane, or in a chloroplast. So the actual amount of energy that's allowed depends upon the bond structure of the molecule, which can change depending upon its environment. It can also change depending on its temperature.
That changes like where it's located.
Yes.
What about width?
We're getting there.
Oh, sorry.
Yeah, no, good. That's exactly right, because this doesn't look much like a chlorophyll absorption spectrum yet, does it? All you have are the peaks. And those peaks are associated with the two allowable electronic states. Okay? And the magnitude of them has to do with the probability that you will go from this state to this state. So the intensity of absorption is dependent upon the probability that the electron will move to the higher energy state. Okay. Okay. Talk over myself. Okay. Each one of those electronic states, or you might think of them as shells, although people don't like to use that term, but I think conceptually it helps to make sense. So if the electron moves to the higher electronic state, within that state, there's also other allowable levels of absorption due to the fact that maybe the molecule is not at absolute zero. It has some temperature, it has some vibrational levels. And so you can go from the ground state, and if it's slightly warm, the molecules may be in this a little bit higher level. So the amount of energy between here and here, that is the energy equivalent of an infrared photon, a thermal photon. So nanometers, or microns, as opposed to nanometers in wavelength. And so this might also be a couple of microns, a wavelength equivalent energy. So if the molecule is high energy, it's vibrating, you have higher, you might find yourself in this vibrational state of the ground state, and then you could absorb potentially this much energy, or this much energy, or this much energy. Again, it's quantized. So within each electronic state, there's some vibrational quantized states. If you start looking at the difference in energy associated with movement between this electronic state and this electronic state within these vibrational levels, that starts to give you absorption at quantized differences here. Okay?
So is E1 0 Kelvin?
It's the ground state of whatever your ground state is. So it sort of would depend on,
The rise?
it could be absolute zero, it could be whatever you started off in your lab, right? And then you're looking at everything relative to that. It could be at 20 degrees, and you could raise the temperature, or the fact that you've got it in a fluorometer could be warming up, right? So it's relative to that. The people who do this usually try to do it at very cold. So the likelihood that you go from any one of the vibrational levels in the ground state to this range of vibrational levels in the second excited state, the probability that that's likely to occur is what gives rise to which is the peak and which is a little bit lower absorption. Okay. The greatest probability is occurring between the ground state here and the ground state here in this example. Okay. It's not always true. Sometimes there's the preferred would be over here, and you'd have lower vibrational levels with lower absorption coefficients because of lower probabilities. And we can also have different vibrational states for the second electronic state. Okay. So now we're starting to see some spread here. Yeah, okay. Then, within each vibrational state in each electronic state, there are rotational states. And those are also quantized. And each one has an associated probability of occurrence. And so if you can have a molecule that's in its ground state second vibrational level, and maybe, or say first vibrational level, second rotational state. And there's a probability that it will absorb energy of this magnitude, and that will give you one of these peaks here. Okay, so now you start to see this smooth curve. If you look at chlorophyll, it tends to look a little bit more like this. These are the electronic states. These are the vibrational states. And then within the vibrational states, you'll have the rotational states around them. Okay? So that's what's happening at the molecular level. If you look at something like a carotenoid, and Ivana's going to talk more about pigments, and you look at the absorption, sometimes you find there's a little more symmetry, where this is the electronic state, and you have a higher energy and a lower energy vibrational state. And again, it just has to do with the probability or the likelihood of absorption of any one of those quantized energy levels, which is associated with a particular wavelength. Okay. Okay. All right. So chlorophyll has the two electronic states. One's associated with the energy of a blue photon at about 443, and one with a red photon at about 676. And again, it depends on if it's in the cell or if it's in extract. This is the chlorophyll molecule. The absorption occurs in this part of the molecule, in the porphyrin ring structure. It's got these really nice chiral rings. There's 4 of them. There's magnesium in the center. The carbon tail is what roots it into the membrane of the chloroplast. But this is the part that sort of sits out like a plate for absorption. And in my biochemistry class when I was in graduate school, the professor said that, and I have never been able to verify this, so if anyone can, let me know, said that it made a difference if the light was coming in, propagating this way into the molecule versus coming in this way. And that it was one direction which there was absorption in the blue, and the other, it was almost as if there was like a polarization sensitivity, and the other direction was responsible for absorption in the red. I don't know if that's true. If you find out that it's true and you find a reference, let me know, because I haven't been able to verify that, and he's no longer around. Wow, thanks. I forgot to turn my own phone off. Okay, so the other way to look at this is, again, here's the energy spectrum, and here's the absorption associated with blue and red. Here is a way of looking at the absorption as a function of energy, which is also wavelength. Higher energy blue, lower energy red. And so here's your absorption by chlorophyll in the blue and in the red. And when you lose energy from this higher excited state down to this lower state, that energy loss is due to heat. And we'll talk more about this when we talk about fluorescence. You can also lose a little bit more heat when the electron is going from this red electronic state. But in addition to losing energy, the molecule losing energy as heat, it can also lose energy as a photon. And that is also quantized. And that photon that it loses has this much energy equivalent, which means what color? Would it lose a blue photon? No, because this is a blue photon. That's how much energy is a blue photon. So what color is fluorescence?
Red.
It's red. And you know that because you know that chlorophyll fluoresces red, right, from fluorometers. But this is why. Because it's going from an energy loss from that first excited state to the ground state. And that amount of energy is a red photon. Okay? So we'll talk a little bit more about that next week. All right, so there's chlorophyll A, there's the absorption by chlorophyll A. It's color-coded by wavelength. We call the blue peaks the Sorey band, the red peak the Q band. There's the structure of the molecule. One of the things to keep track of is that chlorophyll degrades, and it can degrade into degradatory pigments, and Ivana's probably going to cover more of this, but I want you to be thinking about it now when we talk about absorption, because it's going to impact the way that we think about absorption. If it loses its tail, this part, the absorption spectrum changes from this to this. There's some slight changes in the peaks and the relative heights. If it loses its magnesium in the center, because of heating it up or letting it get acidified, then its absorption changes to this, which again is slightly different. If you just lose the magnesium, you get phaophytin, and here's your absorption spectrum. We will be doing a lab where we measure chlorophyll concentration by dissolving phytoplankton into acetone. If your sample heats up, you lose the magnesium, and then you will no longer have chlorophyll in your sample. So you're not going to do a very good job of measuring chlorophyll. Similarly, if you blanch it or heat it and add acid, you can get these various derivatives. And so you're not doing a good job. So this is just the little, don't let your sample warm up, protect it from light, protect it from heat. Interestingly, when phytoplankton are eaten by zooplankton have acidic guts like we do. And so what happens to the chlorophyll? It loses its magnesium and becomes phaophytin. So that's a challenge. But it's an opportunity, because what it means is if you measure a lot of phaophytin, it suggests a lot of grazing. So by looking at the changes in the optical properties of the pigment, it can tell you something about the ecology of what's happening. So,
is that the only process that does that? I mean, is it a one-to-one correlation between the amount of that and the amount of grazing, or are there
it's a qualitative proxy. I'm sure someone will come up with a relationship to a more acidified ocean and more phaophytin. I don't know. But if you have very high phaophytin, you know that the chlorophyll has undergone some degradation. Yeah, Ivana.
There's different types of metabolites of the chlorophyll. There's quite a lot of papers out there that is looking at different types of metabolites and trying to connect them with different processes. For example, the cutting of the tail is done by that, but then you can lose the magnesium from the ring itself. The modifies with different metabolic attachments, pigment that is indicating something else. For example, the rupture of the cells or dying of the cells. You have to have certain enzyme that does that. There's this whole group of people who are studying these metabolites of the chlorophyll and trying to connect them with the stuff. And I could put some papers.
So when I was a graduate student, the range was to use phaophytin as an indicator of grazing. And somewhat that fizzled out because people realized that it could happen, it could be oxidized on your filter.
Right, that's what I was just going to say. Yeah, you have to be really careful.
You have to be very careful in interpreting what might have caused what you're observing.
Mm-hmm. And so a lot of people would coat their filters with magnesium sulfate to keep the pH on the filter pretty basic, so that your filter wouldn't be acidifying the chlorophyll before you got a chance to measure it. So.
It's really the importance is, this will not happen if you kind of follow what we tell you to. No.
Right.
If you think about it, all these things that Collin has mentioned, you want to filter in low pressure, filter so they don't rupture the cells. You want to really try to preserve the samples as neat as you want. You pigment analysis right away and look at nitrogen that is an oxic environment, deep cold environment that stops any kind of degradation. So you can take the steps to ensure that what you're actually seeing in your pigment data.
Is real.
It's actually real to the most extent. If we don't figure out later on that they're completely messed up or something, but for now it's real.
Mhm. And as much as you can do in situ, the better. The less handling that you have, the better. Yeah.
Yes, is this why also bioflows measure at 412 nanometers? Is fighting one of the reasons?
Well, pharophytin actually peaks bluer than chlorophyll does. I think they're measuring 412 for Sedum. Yeah. And they're measuring the fluorescence in the green, where presumably phytoplankton are not fluorescing due to excitation at 412. If you measured red fluorescence due to 412, you might be able to maybe pull out some information about pharophytin. Pharophytin also has a much lower fluorescence efficiency than chlorophyll, so it's harder to measure, but yeah.
I have this book, I'm going to leave it here for the class. If you're really interested in phytoplankton pigments, you don't have this book, feel free to learn everything about it. Every
Great book.
single thing is in there, so it's all yours.
It's fabulous. Okay. So, what are the major absorbers in the ocean? We're going to start with what? What do we have?
Water.
Water. Major absorber. What else? Phytoplankton. Sedum. Bunch of what? Everything else. Dead stuff. Living stuff that doesn't have pigments. Minerals. Okay. All those things. All right. So let's look at them. So this is a series of 3 different absorption spectra that I measured in 3 different environments. The open ocean is the absorption in the blue line. The coastal waters is this one. And a really productive estuary is this one. And so what do these 3 environments all have in common with respect to their absorption? Water. And what is water doing?
So it's red and
So they all look really similar out here because water's dominating, right? And they all look really different here because all the other stuff that absorbs tends to absorb in the blue and also tends to vary globally in huge concentrations. Okay, so they're all strong red, they differ, da, da, da. Okay. All right, so helpfully, thank you, Mother Nature, absorption is a conservative property. So it turns out that if you measure the absorption by all the different constituents in the ocean, you can add them up and get the total absorption spectrum. So you could take the absorption by water, and you could add it to the absorption by all of the different dissolved compounds, and you could add it to all the absorption by all the different particles, and you would get the total absorption. And you could do closure. Okay, by measuring the total absorption and measuring the sum of the absorption of all the parts. It also turns out that absorption is proportional to the concentration. So if you had one constituent and you had 10 of them, you could measure the absorption of one of those constituents and multiply it by 10. So the example that we use is if we look at the absorption due to chlorophyll, and we say, well, this is how much 1 molecule of chlorophyll would absorb. If we measured the concentration of chlorophyll, we could just multiply it by chlorophyll's absorption and get the total absorption by chlorophyll. So we will do a little example of Beer's law in the lab tomorrow. So the problem is it's really impractical to measure the absorption of every absorber in the ocean. You know, you'd have to measure all the phytoplankton, all the zooplankton, all the bacteria, all the pieces of junk, the dissolved molecules, the sediments, and that's a huge job. So we tend to group components by their common absorption spectra. And that way we can just break that into the major groups. And so the major groups that we tend to divide it into is water, dissolved organic matter, colored or chromophoric dissolved organic matter, phytoplankton, which sometimes we do with a phi, sometimes a pH, sometimes a pHi, sometimes a pHyt. This is a phytoplankton here. And then something called non-algal particles, which means everything that's not phytoplankton. And I'll tell you, we'll discuss what's actually in that that component. Okay, so here's the absorption by water. This is on a linear scale from blue to red, the absorption in units of per meter. This is on a log scale. This is from Pope and Fry's 97 paper. There's a nice but sort of dated compendium at this website, which I think you have on the lecture site, where you can go and look at the history of the absorption by pure water. It's not as simple, it has not been as simple to measure pure water as one might think. Here's an example from Scott Pegaw's paper where he showed all of the absorption spectra for pure water for a variety of different labs, a variety of different techniques, and then discusses why they're different and what their strengths and weaknesses are. I think it's really worth doing. You notice they all do pretty similar in the red where the signal is strong. They do poorly in the blue where the signal is weak and where any contamination might occur. It's really hard to make pure water and keep it pure. It's really reactive once you make pure water. It tends to grab things off of glass or plastic or whatever container, or grab things out of the air. So one of the things that Scott Pegaw and Ron Zanefeld worked on early in the 90s is looking at the absorption coefficient as a function of temperature. And it turns out that the absorption at this region here is lower when it's cold and higher when it's warm. How can we relate that back to what we talked about 10 minutes ago? What's happening to the molecule as you change the temperature from 5 to 30 degrees? It vibrates. So if it's vibrating, what does that mean in terms of its ability to absorb quantized energy? It's going to change, right? So the fact that it's vibrating changes somewhat the probability of absorption in a vibrational harmonic. This is a vibrational harmonic for water. And it's a lot. I mean, that's a pretty big difference. 0.1 per meter is significant. And in fact, if you look at the absorption at 740 nanometers as a function of temperature, you see that it increases. And the reason why this is really important is because we use pure water as a reference when we're measuring absorption with in situ absorption meters, or in a spectrophotometer when we're trying to measure Sedum absorption. We always have to have pure water as a reference. So it makes a really big difference, the temperature of your pure water and the temperature of your sample. And you can get huge differences here. Another way I like to think about it is that water molecules, this is from an intro oceanography, I'm sorry for the quality of the figure, but each of these is a water molecule, is the oxygen and the hydrogens. Water molecules cluster together and are held together by hydrogen bonds. And though the size of those clusters of molecules depends upon the temperature. When it's really cold, the molecules are not moving as much, and they tend to form hydrogen bonds between the sort of partial negative charge of the oxygen and the partial positive charge on the hydrogens, and they tend to bond together and form big clusters. When it's warm and they're vibrating, the size of those water clusters changes. And so you'll see a change in the number of water molecules in a cluster as a function of temperature, which is also going to change its scattering properties. And so when we talk about scattering, by, if you're getting scattering by large clusters versus small clusters.
So figure A, that there was a linear relationship between temperature and absorption.
Right here, yeah.
So
Yeah.
the 1A, that's for 740.
Yeah.
Is that the case? Is it linear for all wavelengths?
Yes, but for some wavelengths there is no temperature dependence because it's not a harmonic. But it happens to be a really strong harmonic. There's a slight harmonic here. It's not as big. Not here. This one's really big. This happens to be the area, of course, where you'll find out, this is the wavelength band that we use to scattering correct absorption measurements, because absorption by other materials is so small in the infrared. But absorption by water and the temperature dependence is huge, so you have to correct for the temperature difference in order to then be able to use this region of the spectrum to make other corrections on your measurements. Turns out if you add salt, it also makes a difference. So if you add, the more salt you add, the lower the absorption. Wait, is that right? No, the higher. Yeah, higher salt, higher absorption. And again, if you think about what's happening, the salt is getting pulled apart by the water molecules, and the water molecules are clustering around chlorines and sodium. And so there's you will make use of these correction factors which give you a spectral dependence of absorption based upon the salinity of your water. So if you're measuring in a lake versus measuring in the ocean, you have to think differently about how you're correcting absorption, because absorption by pure water isn't constant. All right. Dissolved organic matter, or actually this is dissolved inorganic matter. So we get pretty fast and loose with the term Sedum, color dissolved organic matter. If it passes through a filter, it's Sedum. Well, it's not just organic material that passes through a filter, it's all the inorganics too. They tend to absorb in the UV. So bromine is one of the dominant absorbers. It's also one of the major contributors to salinity. Hydrogen sulfide, nitrate, this is the absorption by nitrate. This is why we make use of the fact that we can measure nitrate concentration optically in the ocean. It's by measuring the absorption coefficient in the UV. Here's sulfate. So a lot of these inorganic salts have a pretty strong absorption coefficient in the UV. Again, an opportunity. You can build a sensor around that. The dissolved organic matter, Sedum, we typically take a water sample, we filter it, collect your filter for a different analysis, and then you measure the absorption in a cuvette of the dissolved fraction. And it tends to be exponential in nature. It can vary a lot in terms of magnitude. More Sedum, more absorption. This is a really nice figure from Heidi Dearson's paper in 2006 showing, actually I think Grace Chang at UCSB took this picture. It's of her paddle in really high Sedum water, and you can just see that really, really strong blue absorption and shifting the light reflecting out into the red. And of course there's a nice Sedum wave. Right.
Should be pointed out, the paddle is white.
Yes, I'm sorry, yes, the paddle is white, and so you're seeing the really strong color of the water. So if you read some of the older papers, they'll talk about Sedum as Gelbstoff, which is German for yellow substances. I think John Kirk calls it Gilvin, which is,
Pretty.
yeah, it's prettier, but it still just means yellow stuff. And it's because Sedum is always in water. And so if you put the water absorption coefficient on here, the minimum absorption is somewhere over here in the yellow. Okay, so this is the same paper from Kirk, from some inland waters in Australia. And it turns out that people tend to model the spectrum analytically with just an exponential function. You can fit a function to this curve. You need to know the absorption at one reference wavelength. And if you do that, say if you know the value of that curve at 400, you can predict what the shape of that curve is everywhere, if you know something about the slope of that line. And so this is an older paper by Ken Carter, where he looks at, if you put it on a log scale, it linearizes it, and then you can look at the slope of this, and you can see that that slope changes really significantly depending upon where you are. And it turns out it changes based upon the composition of the organic matter in the water. And so in this paper, it was really nice. He looked at the difference between the ocean water and the loop current and the water coming out of the Mississippi River. This was more terrestrial. He called it fulvic material. This was more marine-derived humic material. And you could see that the terrestrial had a much steeper slope. And so people got very interested in looking at how that slope varied over time and space, because it would tell you something about the composition of that organic matter. So again, you might have a lot of variability in the slope, but it's meaningful variability. And so you can use it to trace water masses. So this is, again, he just put it on a log scale, look at the different slopes. A student of mine took a different approach and actually looked at the slope of the absorption in the equatorial Pacific as a function of depth and a function of longitude. And found that if you took a water sample and you filtered it at 0.2 microns and said everything less than 0.2 microns is dissolved operationally, and you looked at the slope of that material, you found that the average slope was about 0.014, so some steepness in here. But if you took the water that was between 0.2 and 0.7 microns, now why would she pick that? Well, 0.2 is what most people would use to separate particulate and dissolved. But if you're actually interested in measuring the particulate matter, you use a fiberglass fiber filter, and those are 0.7. So you don't have closure if you look at particles bigger than a glass fiber filter and dissolved matter less than a 0.2 micron filter. So she's like, what's in between that? What's in between 0.2 and 0.7? Well, it turns out that it has a much flatter slope and a really different distribution. When you look at the water masses here, this really high concentration of very steep sloped sedum is being upwelled from the deep waters under the equator. And this fraction, which I would call colloidal, 0.2 to 0.7 submicron, but not really dissolved, has a much flatter slope. So a slope that was more like this, and was associated with fresh phytoplankton blooms. So that begins to suggest that whatever is in this dissolved fraction, whether it's 0.2 or 0.7, can actually change depending upon its origin and maybe the processes that have been happening to it. Is it really old and refractory and has a steeper slope? That's the steeper slope that Carter found in the Mississippi River for terrestrial fulvic material that's probably been washed off of the landscape and into the Mississippi, compared to fresh organic matter that's recently broken down. So the spectral slope contains a lot of information. So it behooves you to measure the absorption by Sedum very accurately so that you can derive a spectral slope and begin to look at trends in that slope to tell you something about the origin of that material, where it comes from, how much it's been degraded, and what its spatial distribution might be. So we'll be doing that tomorrow.
What wavelength would you use to derive this slope?
Well, so that's a question. Some people go from 200 to 500, some people go from 300 to 500, some people go 400 to 600. So we'll talk about that. Because it's different material. In the dissolved fraction, as you go further to the UV, you tend to be more sensitive to nucleic acids. So where you calculate your slope, I calculate 3 different slopes. And I do them all the time, and then I compare all of my 400 to 600 nanometer slopes with each other. I compare all of my 220 to 300 slopes with each other. Yeah. And you can tie it to, that way you can also tie it to the slopes that are derived from marine chemists who are using it to determine the number of benzene, like the number of ring structures that you have. I've just lost the word for what that is. But, so, yeah. So, when I took this class in 1987, I measured a lot of absorption coefficients in Puget Sound, and I published a paper on it from my class project. So, be inspired. Well, I published it like 2 years later, but anyway. So, in the meantime, I went around and I digitized everybody else's paper with absorption spectra of dissolved matter, because nothing was electronic back then, it was the Dark Ages. And then I calculated the average slope of the Sedum. And I found that the slope was about 0.016 or 0.017. That described the steepness of that absorption. In 2003, Marcel Bavin went around the European coastal waters and collected 346 samples, determined the spectral slope of the Sedum for all of those waters, did a histogram, and it turns out they're exactly the same within the uncertainty that we both published. So the robustness of the spectral slope is pretty strong, because this is digitized data from the, here's 1966, right? So even with the techniques that were being used in the 60s, you could derive a spectral slope compared to modern technology in different waters. And so the spectral slope is robust, but the variability within it is meaningful. Okay. So now we go to the other part of this process where we filter, and we collect a filter, and we have particles on that filter, and then we measure that in a spectrophotometer, and we'll be doing that on Thursday. So you take your filter, you've got particles on it. Has everyone seen a glass fiber filter with particles on it? Has anyone not seen it? You get to see it this week. Okay, so you filter your water sample, you end up with particles on there, it's a certain color. You begin to figure out what's on the filter by looking at the color. Like, diatoms, or a lot of tidal matter. And then you measure it in spectrophotometry. You look at the transmission through it as a function of wavelength. You get a curve that you might even be able to identify a phytoplankton peak, even here, but there's a lot of other stuff in there. You take that same filter and you put it back in your filter cup and put methanol on it. And methanol goes into the cells, removes the pigments from being bound to the thylakoid membrane, and then you wash it through. And what you're left with are all the particles as they were on the filter, including the phytoplankton, minus the pigments. You can even look into the microscope and see the intact cells, because methanol is a relatively mild solvent. And you measure it again, and you get what we now call NAP, or non-algal particles. And you get a spectrum that looks something like this. What does that look like to you? What kind of analytic function might you fit to that? Exponential. Hold that thought. Okay. That is the absorption spectrum of particulate matter in the absence of pigments. So it's everything on the filter minus the phytoplankton pigments. So, detritus, dead organic matter, bacteria, viruses, zooplankton, polysaccharides, any particulate matter minus the pigments. And it has an absorption spectrum very similar to Sedum. Okay. If you take that scan and you subtract it from your first one, total particles, and then just the particulate matter minus the pigments, what are you left with?
Pigments.
The pigments. Well, why didn't you just extract the pigments and measure them in a vial?
Very challenging to do that.
Turns out it's easier. But it's a good guess. But if you measure the, what?
Bowls for closure?
It's what?
Do bowls for closure?
Oh, you could do both for closure.
But then you lose the outcome back, right? No.
You lose the pigments as they were in the cell.
Yeah.
You pull them into an extract, and now they're all floating freely, not attached to each other, so their absorption magnitude, the maximum might change a little bit with wavelength. They're not bound together, they're not bound to proteins. And so by doing this, it allows you to reconstruct the absorption by the pigments as they were packaged in the cell. It is a brilliant, simple approach. that Motowaki Kishino published in 1985. And it allows you to derive the absorption by phytoplankton intact on a filter pattern. However, it includes phaopigments. If there's a fecal pellet on there that has phaophytin, that will get extracted and it will be added. It will be included in this phytoplankton spectrum. So if you have really high detrital particles in there, you may overestimate your phytoplankton absorption. But you can do some uncertainty measurements on that. This is a healthy culture, which shows that there's relatively little contribution of cellular material to a healthy culture. But we would call this the non-algal particle component of the phytoplankton. So it includes the phytoplankton structure, just not the pigments. All the pigments show up in here. Okay, so you'll be doing this method this week. Let's see, microphotometry. Rodolfo Turriaga and Dave Siegel, who has a student in the class, they did a really interesting approach where they took a spectrophotometer, which is measuring, again, light coming in, light coming out, and they focused it on a microscope down to a single phytoplankton cell and looked at the absorption of individual cells within a sample. Here's one sample from the Sargasso Sea, and these are all the different phytoplankton in that one sample. Here are, oh, this is another sample. Okay, I can't remember which is which. It's literally this blurry in the old journal. And then, they also did it for detrital particles. And so then the point is, of course, if you add each one up, you get the total absorption by phytoplankton for that sample. It just took like a day to do one sample. So it's amazing to think about from the perspective of look at the variability between different phytoplankton within one sample. But whew, yeah, sometimes it's better just to group it together and call it good, get a standard deviation. So, but you can measure individual cells. Let's see. Here are different species of phytoplankton, and this has just been scaled so you can just look at the variability in shape. And so you can see there's an overriding feature of phytoplankton absorption, the two peaks of chlorophyll, some other pigments in here. This would be the average of all those spectra. If you look at the variance spectrum, you see these very nice features in variance, right? What do these features tell you? Differences between different taxa. And what is doing this?
Pigment composition.
Pigment composition. So when you see differences in the shape of these curves, it's because they have different pigment composition. Now, that can vary between taxa. Diatoms and dinoflagellates certainly have different pigments. Diatoms grown under different conditions have the same pigments, but they may have them in different ratios. So the variability in these parts of the spectra are really informative in terms of species composition and also within a group, a taxonomic group, of maybe their photoacclimation history or how they've made different pigments in response to light variability. Yeah.
So the first figure is normalized spectra, right?
Yeah, I just normalized it to the area under the curve. And so you're just looking at differences in shape. And so you can see some have relatively low to high blue to red peak ratio. They have really different carotenoids. There's this difference in chlorophyll B versus not chlorophyll B. A lot of carotenoid differences in here, chlorophyll C's, accessory pigments. And so each one of these is different. This peak right here, this variability in essentially the width of that peak, that's phaophytin. Phaophytin absorbs a little bit redder, and so if your peak is wider, it means you have a little bit more phaophytin in your sample. And so by doing this sort of scaling, you can begin to identify which pigments are really variable in your suite of samples. It's a really quick way of doing it. And you can just identify all of the different pigments that you might find. These are phycobilly pigments associated with cyanobacteria and rhodophytes. These are the different chlorophylls and carotenoids, but they're really, and Ivona has the paper, and or has the book, and she'll talk about the specifics of these pigments, but they're really easy to see when you start looking at phytoplankton spectra. So, okay. No way.
Well, you can stop wherever.
I'm
you
stopping here. All right, so we'll return after lunch to talk about pigment packaging.
We're back at 1 or 1:30 or what?
We're back at 1. We can do 1:30 actually. Whatever you feel like. Was it 2 days?
It's a lot.
1:30. But remember, you have homework. You have to sit with a classmate and go through those.
Oh, I only have 8 slides left, so there's only 8 slides. Whatever. So 1:30 is fine. I'll be quick.
1:30. I mean, we have just one lab today. There's a lot there too.
And it's a fun lab that you don't have to report on.
Exactly. The one lab you don't have to report.
Exactly.
It's the one to get your curiosity
Yeah.
firing. But remember to sit with one of your classmates and answer, if you want, I can put this question back. You guys will come back.
Something
Otherwise,
from
you
the
can get
past,
it from
something from the
website.
future.
You have to make sure. You stand here as couples, and the faculty will do the same. And you introduce each other. So here it is. And it can be fun. Don't stress about it. Just an opportunity for us to get more to. And you have access to it also through the website. So everything that you're seeing is being posted there. If you forgot something or didn't write something, it's all of it. So we'll have lunch at Brooks Hall, and then 1:30 back here.
It's fine to leave our stuff here.
It's fine to leave your stuff here. So I've never got anything stolen so far in Maine. It does happen sometimes to some people. I've never had any issue. So it's I mean, we don't lock our cars. We don't. So it's. But you never know, it might hit you. But I'm going to leave my computer and hope that nobody's going to decide to adopt it.